Hello everyone, I'm Taejun Ham, a postdoctoral researcher from Seoul National University. In this talk, I'll be presenting our recent work titled Genesis, a hardware acceleration framework for genomic data analysis. This is the work done in collaboration with my colleagues and collaborators at Seoul National University, UC Berkeley, Sungkyunkwan University, and Duke University. As many of you know, DNA is the chemical compound containing the instructions an organism needs to develop, live, and reproduce. As shown on the right, DNA is made of two paired strands, where each pair is represented with a single character that corresponds to one of the paired nucleobases A, C, T, or G. And the DNA sequencing, or genome sequencing, is a process of identifying the base pair sequence of a DNA. DNA sequencing or genome sequencing is widely used for various purposes. A person's genome sequence can be analyzed to check if the person is particularly susceptible to a specific disease or to identify the exact type of cancer. For example, genome sequencing is often used to identify the exact variant of lung cancer, which enables the use of specific targeted therapy. Genome sequencing is used for COVID-19 researches as well. Performing sequencing on COVID-19 virus allows researchers to identify and reveal many variants of COVID-19 viruses. Genome sequencing was very expensive in the past. For example, the Human Genome Project started in 1990, took 13 years, and cost $2.7 billion. However, the cost of genome sequencing has rapidly decreased over the last 20 years due to the advancements of the wet lab sequencing technologies. Nowadays, a commercial genome sequencing service only costs about $300 to $700, and the throughput of a single genome sequencing machine exceeds 1 genome per hour. Such a huge reduction in genome sequencing cost resulted in the rapid increase in the amount of genome data. Naturally, the computational demand to process such genome data has sharply increased as well. Genome sequencing instruments do not directly provide the entire base pair sequence of the genome under study. Instead, it provides a set of fragmented genome sequences that need to be processed with the various algorithms to construct a single genome sequence. The process of identifying the single genome sequence and some additional metadata is called secondary analysis. Secondary analysis consists of many different algorithms. The exact process heavily varies according to the goal of the analysis and the type of genome sequencing instrument used. Our work mainly focuses on the GATK4 best practices secondary analysis pipeline, well suited for the popular Illumina genome sequencing instrument. Specifically, we target the data preprocessing phase of this pipeline. The data preprocessing phase consists of 4 stages, and the runtime breakdown shows that complex stage such as alignment takes most of the runtime. The importance of the genome sequencing and the fact that alignment takes the majority of the genome sequencing time motivated computer architects to design the hardware accelerator for the alignment. For example, Darwin published at SPLOS 2018 achieves 15,000 times speedup over software run on CPU platforms for long reads produced from Oxford Nanopore Technologies. And GenX, published at ISCA 2018, achieves over 200 times speedup for short reads produced from Illumina instruments. And there are many other recent hardware accelerators targeting specific algorithms dominating the runtime in other phases of the genome sequencing pipeline. This advent of the hardware accelerators targeting the complex algorithms in the pipeline shifts the bottleneck to remaining simple data manipulation operations such as the ones shown in orange boxes. For example, if we assume GenX alignment throughput to process the alignment stage, the alignment after acceleration only takes 0.7% of the total data preprocessing phase runtime, and the orange-colored simple data manipulation operations now account for 93% of the total runtime. It is natural that developing efficient hardware accelerators for complex algorithms requires significant effort. However, our idea is that developing hardware accelerators for relatively simple data manipulation operations should not require a significant effort. Unlike monolithic accelerators targeting complex algorithms, such as Darwin or Genix, Genesis provides a way to design the accelerators for data manipulation operations in a composable way by leveraging the fact that such data manipulation operations are often a sequence of common primitive operations. With our framework, a user first utilizes the Genesis SQL frontend to represent the target data manipulation operations in a way that can be easily mapped to the hardware. Then, the components in the Genesis hardware library, containing configurable accelerator building blocks, is used to construct the data flow pipeline for the specified SQL query. Finally, the Genesis backend automatically augments the hardware pipeline with parallelism, deploys it on Cloud FPGA, and allows a user to access it with a high-level API. Here's the brief outline for the talk. Moving forward, I will talk about the components of Genesis framework, present evaluation results, and conclude. A key intuition behind the Genesis framework is that most genomic data manipulation operations can be easily represented with one or more SQL queries. A few prior work also share this observation. For the data manipulation operations in the genome sequencing pipeline, there are two key data types, references and reads. A reference is the reference genome sequence for an individual organism of a species. For example, Genome Reference Consortium regularly publishes the Human Reference Genome Sequence. A read is a fragment of the genome sequence measured using sequencing instruments. For our work, we target data manipulation operations after the alignment. Such reads contain metadata representing the alignment information produced by the alignment algorithm. At the bottom of the slide, we show examples of the reference sequence and 3 aligned reads. Near 1 billion reads are utilized for the human genome sequencing. One thing to note is that these reads are not identical to the reference sequence. Reads may contain different base pairs for the specific position, as shown in red-colored characters, and reads may also include insertions or deletions. The key data type in genome sequencing, references and reads, can be conceptualized as a very large relational database, and they can be represented in a tabular form as shown in this slide. And data manipulation operations can be represented as one or more SQL queries. Since the reference genome sequence is very long, it is partitioned into multiple fragments, and each fragment is stored as a row in the table. For conciseness, this slide only shows a single row of the reference table. More detailed explanations are presented in the paper. Reads table contain many rows where each row represents a single read. Each aligned read also contains alignment-related metadata such as pose or cigar. Pose is the aligned position of this read, and cigar is the special metadata that contains information on insertions or deletions. For example, a 2M represents that the first two base pairs in this read are well aligned. 1D represents that this read lacks a base pair in this particular position. 13M following that represents that the 13 base pairs are well aligned. Genesis SQL Interface supports many standard SQL operations such as select, where, group by, limit, count, and sum. In addition, Genesis supports extra genomic-specific operations post-explode and read-explode. Post-explode is an operation that expands a specific row of the table into multiple rows along with its positions. For example, applying post-explode operation on the row of the reference table results in 33 rows with 2 columns. Each row represents each character in the sequence of the target row, and the post column contains the relative offset plus the original post value as shown in the figure. Popular SQL extensions such as Hive Query Language or Spark SQL also supports this operation. Read-explode is similar to the post-explode operation. However, this operation considers genomics-specific cigar metadata. For example, for read number 2, this operation does not put any base pair for post 14, considering that this read has a deletion at post 14 as indicated by its cigar metadata. Note that read-explode can also handle insertions or other types of cigar operations. Let's say that the user wants to compute the total number of mismatched base pairs between the reference and all the reads in the read table. To do this, a reference table needs to be post-exploded. Users can represent this operation in SQL shown on the bottom left of the slide. Then, the number of base pair mismatches between the reference and each read in the reads table needs to be computed in 3 steps. First, a single read in the reads table needs to be read-exploded as shown. This operation can be represented in SQL as shown on the bottom right of the slide. Second, the outcome of the previous operation is now inner-joined with the subset of the ref table using post as the key, and its corresponding SQL is also shown on the bottom of the slide. Finally, the number of rows that have a mismatch between the ref base pair and the read base pair are counted and outputted. Then these 3 steps are repeated for other reads. The main advantage of using SQL as the front-end interface is that SQL and its column-based executions map very well to data flow architectures. A few prior works such as Q100, Linkage, and SDA also acknowledge this fact and have proposed hardware acceleration techniques for SQL queries. On this slide, I show you how one can stitch the Genesis hardware modules to make a data flow pipeline for the example application discussed in the previous slide. The very first step of the example query is to post-explode a reference row. Since the reference data is reused for multiple reads, this data needs to be mapped to the on-chip scratchpad memory in order to take advantage of data reuse. For this purpose, the reference sequence is loaded from memory using a memory reader module of the Genesis hardware library, and then provided to the SPM updater module along with the constant post value. Then, the SPM updater module stores the loaded value in the scratchpad memory. One thing to note here is that in hardware, the post value for each row is not explicitly stored to avoid redundancy. The next step is the read-explode operation on the read. For this purpose, reads need to be read using 3 memory readers, each reading a column of the reads table. These loaded values are then fed to the Genesis read-to-basis module that performs the read-explode operation. The outcome of the reads-to-basis module is then fed to the inner joiner so that they are joined with the reference using the post as key. Another input for the inner joiner is the reference. To obtain the reference, the post value of the read is provided to the SPM reader module, which loads the corresponding reference from the scratchpad memory and provides it to the inner joiner. The final step is to count the mismatch between ref_seg column and read_seg column. For this purpose, the output of the inner joiner is passed to the filter hardware module, which filters out rows where the reference sequence and the read sequence match. The output of the filter module then only contains the mismatched rows, and the reducer counts the number of such cases and passes the final value to the memory writer, which stores this value. Genesis pipeline can be configured to operate at a specific granularity. For this particular example, the pipeline is configured to operate at a row granularity. Such fine-grained pipelining enables all modules in the pipeline to efficiently work in parallel. This process of translating SQL queries to the hardware pipeline is currently manual, but we envision it to be automated in the future. The Genesis hardware library also contains other modules that are not utilized in the previous example. Overall, the Genesis hardware library includes 4 types of hardware modules. First, the Genesis hardware library contains data manipulation modules, which are shown on the right. The joiner, filter, reducer accelerate relational algebraic primitive operations, and a combination of these modules can accelerate most SQL queries. Second, the Genesis hardware library contains a Genomic data processing module read-to-basis. Third, the Genesis hardware library includes DRAM and SPM read or write modules. DRAM access module supports sequential streaming read and write functionality with the support for prefetch and buffered write. Scratchpad memory access modules support both random and sequential accesses, as well as atomic read modify write operations. Finally, a user can also choose to implement a simple custom computation module if necessary and utilize it when designing specific genomic data analysis pipelines. The hardware pipeline configured in the previous slide is then augmented with the Genesis backend. First, Genesis backend exploits the fact that most row-level operations can be processed in parallel and places multiple replicated pipelines within the FPGA, taking advantage of parallelism. Also, the backend attaches the proper peripherals to the pipeline so that the accelerator supports Genesis high-level user API, which enables the users to control the data movement and computation in the host processor. This API allows users to efficiently overlap the data transfers and the computation within the same pipeline or across the different pipelines. Details about this API calls are explained in the paper. With Genesis, we designed 3 accelerators targeting 3 data manipulation operations in the data preprocessing phase of the GATK4 genome sequencing pipeline. Specifically, we accelerate a quality score reduction computation in the mark duplicate stage, the metadata update stage, and the covariate table construction step of BQSR stage. These 3 stages account for more than 80% of the total data preprocessing phase runtime, assuming that the time-consuming alignment stage is hardware accelerated with the GenX accelerator. Due to time constraints, I will omit details of what each stage does and the process of how to construct Genesis-generated accelerator architecture for each of these in this presentation, but we encourage the audience to read the paper. Now I will go through the evaluation results and conclude the talk. We deployed Genesis-generated accelerators on an AWS EC2 F1 instance with a Xilinx VU9P FPGA. For the software baseline, we utilized an AWS EC2 instance running 16 threads with Intel Xeon Platinum CPU and high-performance SSDs. Genesis obtains 2 to 18x speedup across 3 different data manipulation stages. The runtime breakdown shows that the Genesis speedup on the mark duplicate stage is limited by the unaccelerated software portion. On the other hand, the metadata update and the BQSR performance are partly limited by the host FPGA data transfer time. We expect the speedup to improve in the future with a better interconnect between the host and the FPGA that provides higher bandwidth. Finally, note that the speedup can be further improved with the use of multiple FPGAs. The use of Genesis-generated accelerators not only provides performance improvements, but also provides cost savings. The cost saving ranges from 2x to 15x across workloads. This is generally proportional to the speedup, but it is slightly less than the speedup since AWS charges little more for the instance with the FPGA. The right bottom graph shows the FPGA resource utilizations of the 3 Genesis-generated accelerators. We observed that these resource utilization profiles differ. For example, mark duplicates and BQSR mostly utilize combinational logic blocks, while the metadata update utilizes a large amount of VRAM. This indicates that it is possible to co-locate different accelerators on the same FPGA to increase resource efficiency. To conclude, we present Genesis, a hardware acceleration framework for genomic data analysis. Motivated by the observation that most genomic data can be conceptualized as a very large relational database, and genomic data manipulation operations can be represented using extended SQL, Genesis presents a way to effectively design accelerators for data manipulation operations in a composable fashion. Specifically, Genesis leverages accelerator reuse by providing a hardware library that contains modules necessary to build a data flow pipeline for the SQL query and a backend that deploys the data flow pipeline on cloud FPGA. Along with prior works targeting complex algorithms within the genome sequencing pipeline such as alignment, Genesis can reduce the runtime and the cost of genome sequencing and advance precision medicine. Thank you for listening, and feel free to ask any questions on the discussion board.